Dr. Montalvo Ortiz is an assistant professor in the Department of Psychiatry at Yale School of Medicine and research scientist at the National Center for PTSD in Connecticut. Her research focused on the genetics and epigenetics of psychiatric disorders, particularly in Latin America and underrepresented populations. Dr. Montalvo, the floor is yours.
Thank you so much. For me, it's a pleasure to present alongside many experts in the field from Latin America, and for us to talk about our work at the Latin America Genomics Consortium, and that I hope it can promote and facilitate future collaborations. And thank you so much for the invitation. So I'll be presenting our work on neuropsychiatry genetics in Latin American populations. Here I'm presenting what has been a rapid growth on the genomics of psychiatry disorders within the last around 10 years. On the left, I'm showing a plot where we show the number of cases included in GWAS studies from the very beginnings in lighter green, and the projected numbers. And we know that for many of these psychiatry disorders, we have even surpassed those expected sample sizes for genome-wide association studies, allowing us then to identify genetic variants associated with disease risk. So on the right, I'm showing a plot summarizing this status in the field in terms of genetic risk loci identified for many of these psychiatry disorders, plotted by the sample sizes and the number of cases included in the GWAS. And we evidently see that most of the progress has been driven so far by schizophrenia and major depressive disorder. But there's still a lot of work to be done for many of the psychiatry disorders that even when we increase sample sizes, we are still, less than 100 of genetic variants identified. These could be due to a complexity of the phenotypes being identified, the heterogeneity that exists among different cohorts or countries, alongside other complexities in identifying genetic variants in studies that power enough to look at the effect sizes that are needed to identify them. And how does this look across different ancestral populations? We know that our populations, Latin American populations, remain widely underrepresented across the human genetics field. On the left, I'm showing, compared to global populations in billions, the individuals in GWAS studies in millions across time in different GWAS studies. And we see that there's an increase in representation, although this representation has been predominantly dominated by European ancestry, and is in fact disproportionate to the global population of these ancestral group. And when we compare that to other ancestral groups, we see that there is an evident disproportion of underrepresentation of groups other than European ancestry. And when we look specifically into psychiatry disorders, I'm here showing attention deficit hyperactivity disorder, Alzheimer's disease, anorexia nervosa, anxiety disorder, alcohol use disorder, autism spectrum disorder, binge eating disorder, bipolar disorder, cocaine use disorder, major depressive disorder, obsessive compulsive disorder, opioid use disorder, post-traumatic stress disorder, schizophrenia, and suicide and tic disorders or Tourette. So those that are not familiar for psychiatry, just to give you a sense of the different disorders that we have looked at in terms of the representation across different ancestral groups and how this looks like across different ancestral groups, including Admex Americans or indigenous or I would say most of us go into that group, which are Latin Americans. African ancestry, East Asian, European in blue, Middle Eastern, Oceanian, and South Asian. And looking at those in red, which represents those in Latin American countries of Latin American origin, we do see some progress in disorders like major depressive disorder, bipolar disorder, alcohol use disorder, and cocaine use disorder. But for the most wide variety of different psychiatric disorders, their representation still lacking. And this is not yet published. This is very recent data as of this year that will be published soon in the Lancet of Psychiatry and was done by the Psychiatry Genomics Consortium. And I will stress that the need to address this underrepresentation is to happen at every single level when we do human genetics research. This goes from the target population to the assessments that are used, that has to be culturally informative and also provided in their native language that pertains to Latin American populations, for example, that lives in the US are not fully fluent in English. So I need to provide those assessments in Spanish and also in populations across Latin American region that are not necessarily fluent in Spanish, but in their indigenous languages. So start thinking about providing these assessments or diagnostic criteria also in their native language so that we make sure that these are accessible to those communities and that these communities are also well represented in our studies. And also I would say the SNPs arrays that are currently being used to look at the genomic variation at the genome-wide level. For practical reasons, a lot of research groups have used genetic arrays that are not necessarily informative for the genetic variations that is representative of for example, Latin American populations. And these could be solved first by using genetic arrays such as the global diversity array that has been proven to be more informative in terms of the representation of variants that are more prevalent in our populations, and also using technology like whole genome sequencing or the blended genome array or sequencing. Also, the reference panels that are used either for imputation purposes, there's also some gaps that needs to be addressed in terms of the representation of most indigenous communities across the region. And I will say that the development of modeling and statistical methods that allow us to account for the admixture that is representative of our populations is still an area under development, although with some progress, notable progress in that, and thus making us available or permitting us to conduct GWAS studies in our populations that are powerful enough, statistically speaking, that could allow us to identify genetic variants associated with disease risk. And many of these variants often lie in regions of the genome where either it maps to multiple genes or it maps very far away from a gene. So sometimes it's very complex to identify or characterize, I would say, the functionality of these variants. So what are the downstream effects in terms of gene regulation? And for that, we use functional annotation databases, and many of these databases are still generated in samples from European ancestry, so there warrants increased representation in Latin American populations for that as well. And ultimately, our goal is to generate clinical tools that allow us to improve either prognosis or treatment efficacy and overall quality of life of our patients. So the generation of polygenic risk scores is needed to develop more predictive models that are more informative and predicts better in Latin American populations, and that's currently an area, also an evolving area of research. And I will go beyond the research. There is also a need for increasing the representation across the research workforce of researchers in specifically in the South, global South region. And I'm here showing the members of the Psychiatry Genomics Consortium. Though I have to clarify that this is data from over 5 years ago. So this has definitely changed, but still a major underrepresentation, I would say, in nationality and country of origin of researchers in this global South that still needs improvement and engagement for of these researchers in the psychiatry genomics field. And as I said, another complexity of analyzing and studying Latin American populations is of its complex admixture. So here I'm showing on the left plots where we show the ancestral proportions of Native American, European, and African. And we look at how that varies across different regions across the Americas compared to North America, which is predominantly European. We see that for those in Central America, we see more representation of Native American, where, for example, in the Caribbean or the Antilles, we see more representation of the African when we look at different ancestral proportion in our genome. So this complexity and heterogeneity of ancestral backgrounds, it's important to take into account when doing these genome-wide association studies or GWAS. And this is data from the Human Genomic Diversity Project and the 1000 Genome Project, where we look at the different populations across the America, from Mexico, Puerto Rico, Barbados, Colombia, Brazil, and Peru. And we plotted them in a PCA plot, and we see PC1 and PC2. We are starting identifying clusters based on ancestral background. And on the upper plot, we see the reference data sets from European, African, East Asian ancestry, and those in gray, we see the Latin American, Caribbean populations. And digging deeper into those characterized as classified as Latin American, Caribbean populations in the panel below, we see different clusters depending on the country of origin. For example, Puerto Rico clustering between African and European. We have Colombia similarly, and we have, for example, Barbados, mostly African, and countries like Peru, Mexico, and also Brazil, more specifically those indigenous groups around the Amazon, clustering more into the East Asian region. And this is has been recently published in our Nature Genetics paper this year. And I'm here also showing the prevalence of psychiatry disorders across Latin America to have an idea of the prevalence and how heterogeneous it is across the region. And I'm showing the DALYs per 100,000 people. And in the Americas, we see that there's wide variety of prevalence across the region. And when we look at that closely by specific psychiatry disorders, we see that it's the prevalence also, it's different in terms of when we compare it with global rates in high income North America, some of them being more highly prevalent and some of them being less prevalent. Although we have to also notice and emphasize that there's differences in the way diagnoses are being made and how these different mental disorders are being reported. But so far, this is what the data looks like in terms of prevalence. What's the status of the field in terms of GWAS studies performed so far for psychiatric disorders in Latin American populations? So similar to the plot that I showed before, showing number of variants identified by sample sizes. I want to make the note that number of variants here is not going over 25. So there's still a lot of work to be done. It's a huge gap that remains in GWAS studies, and sample sizes of these GWAS studies. Some of them, very few, has passed the 100,000 mark. And this has been possible mainly because of the work of large data sets most of them based in the US, like the Million Veteran Program, the All of Us, and also includes data, those 3 studies of the traits of smoking traits, the alcohol consumption one, and the major depressive disorder. These 3 studies surpassed or very close to the 300 mark, but taking into account that these studies also include data like 23andMe, which is not widely available for research. And we don't really exactly know the representation, for example, of Latin American across the region of the different countries that it includes, but we do know that it's still mainly representing the US. One other example that has not been included in published GWAS so far, but we are actively working on is the MCPS study in Mexico, where we are having 140,000 people all sequenced and data provided for research. And another big cohort that will be upcoming in the next years are a big Colombia effort in missing origin, led by a group at UCLA and Colombia. And these will be also all sequenced and linked with EHR data, and including around 100,000 individuals. So big cohorts are starting to being developed, but not yet accessible for research, and also many of them, for exception of the Colombia cohort, are not very well characterized for psychiatry disorders with the complexity that we need in order to dig deeper into the phenotypes heterogeneity that exists, making it harder for phenotype harmonization when doing these studies. So on the right panel, I'm showing, making the point that most of these studies are coming from the USA, and still there is a huge gap in representation across the Latin America region. Most of at least from the published work in psychiatry genetics, most of the work has been coming from Mexico, Colombia, followed by Peru and Brazil. And among the different psychiatry disorders, most of the genetic variants identified so far has been for substance use traits or substance use disorders, followed by MDD, PTSD, and anxiety. And this again was published in a recent Nature Genetics paper. So in order to address this gap, we established back in 2019 at the World Congress of Psychiatry Genetics, the Latin American Genomics Consortium, with the idea of starting off with psychiatry traits, but also giving space to expand further if there are researchers in our network interested to do so, and that has been the case so far. So from an initial gathering of 25 people, we have grown to 150 members representing 9 Latin American countries, the US, and worldwide, and continuing growing further. And our main goal is to advance psychiatry genomics in Latin American populations by expanding both research and workforce. And we focused on 5 areas: increasing sample size, facilitating recruitment and genotyping, conducting data analyses, um, working on developing novel methods that can account for admixture, and providing educational and training resources. And this consortium was co-founded by Dr. Paula Yussi Rodriguez and myself, and being co-shared also with Dr. Elizabeth Atkinson. So I'm showing here a timeline of how our group has been developing and evolving over the years since 2019, where we established it. So right after the pandemic happened, but we were able to still meet virtually and still work on grant writing and so on. So we focus on obtaining seed funding that allow us to support sample genotyping and support analytical support to do our initial studies, and also working proactively on data access and expansion of our membership. And as we grew as a group, we included different experts across the field with diverse expertise. We then established different working groups, most of these disorder focused, that we'll go over more in detail in the next slide. That allow us to expand our leadership, having co-chairs in each of these working groups. With the one of the policies that we have is that one of the co-chairs has to be based in Latin America, and also having data analysts based in Latin America, and working on outreach activities, and as at the beginning also data analysis. We were awarded the Pamela Sklar Innovation Award at the ISPG, the International Society of Human Genetics, Psychiatry Genetics. And we engaged in training activities, and we've been leading one of the largest so far GWAS meta-analyses of Latin American populations. I will talk about our results shortly. And finally, last year, we've been working on getting out our first set of papers, and we were fortunate enough to having our first published paper this year, and submitted also one of the big grants from the PGC. So these are the different working groups. As I mentioned, these are mostly disorder focused, but also we have one focus on methods development, and that can help establishing pipelines and benchmarking different methods that are coming up for PRS, for local ancestry-informed GWAS, and so on. One focus on molecular epidemiology and functional genomics, and another one more recently established population genomics group. This is where the academic affiliation of our membership is, and these are different countries, including where our membership is from. And this is data from 2021 and 2023. We were interested in looking at how representation looks like across time, across years, in terms of country of origin and country of academic affiliation. And we've been fortunate enough to increase in both of these aspects in our membership. In terms of research areas, our members are experts across different fields and technology and areas of research, and that expertise increased also from 2021 and 2023. Most of them focus or experts in genomics, but also other omics science. And in terms of research area, most of them experts in research related to major depressive disorder, followed by bipolar disorder, substance use disorder, and schizophrenia. In terms of career levels and the gender of our membership, we have pretty equitable membership in terms of gender, and also really nice representation across different career levels, from professorship to all the way down to trainee and undergrads. So these are pictures of our first gathering, which is asked for, joining us for a happy hour at this conference venue. And from that initial gathering of 25 people or so, we have grown substantially. I'm here showing pictures from the meeting in 2022 in Italy and 2023 in Canada. And not included this year's, but the last year's in 2024, it was in Singapore. And we're also interested in how these consortium are facilitating collaborations among our membership. So here we're showing a network analysis where we analyze the publications of our members that were either first or senior corresponding author, just to show publications that are being led by our members. And we look at the collaborations among the different members based on the country of the academic affiliation of our members. And we see a big cloud here, a blue cloud from members based in the US. But we see some connections with researchers based in Mexico, researchers based in Brazil, and we also see some, I would say, islands in the periphery that we hope to unify further. And we're currently expanding this study, but this is so far what we have. This data from 2021 and 2023, and basically showing that we have a bigger unified network, and that's our goal. And we're currently expanding these analyses in our own membership. Showing here collaborators and partnerships of the LAGC, those based in the US and those based in Latin America region, like Mexico City Prospective Study, the Mexican Genomics Database for Addiction Research, Brazilian High Risk Cohort, Proyecto Paisa, Latin Genomes, Neuromex, and so on. And these are our publications so far, and this is also showing the latest publication in Nature Genetics. We've been featured in the news and also in talks readily available in YouTube. So if you want, just do a quick screenshot and you can look at that later. And this is showing the pictures of when we receive our award at the International Society of Psychiatry Genetics, to the three of us, and also Umberto Nicolini and Marcos Santoro from Mexico and Brazil, respectively, who are also part of our leadership. And we wanted to highlight some of the fellowships and grants that have been awarded. Some of our postdocs and main analysts of the consortium have received postdoctoral fellowships, as the case of my postdoc Diana and Jaime from Colombia and Mexico, respectively. And also our one awarded to Diego Roares from Brazil, and a grant awarded to our researchers based in Mexico, Nadia and Gabriela and Carlos. So just want to highlight that I hope I showed the evidence on the representation of individuals of Latin American origin, not only in research, but also in the workforce. And as a response, we established the consortium, where we're expanding collaborations and access to researchers, to researchers based in Latin America, and continuing engaging with researchers across Latin America region, especially those that still lack representation, and continuously also establishing new partnerships in an equitable and sustainable manner. And can someone tell me if how much time I have left?
A couple more minutes, so we can.
Okay, so I'll be very quick. So we focused on alcohol consumption for our GWAS efforts. We were able to include 459,049 individuals, and these are the different countries represented. I have to say a disclaimer that we are included 23andMe in this data set, and the 23andMe represents 200,000 or so. But still, I feel like we did a great job at trying to increase representation across Latin America, but still a lot of work to be done. We started these analyses with just 55,000, but still we were able to see that peak. This is a peak in the ADH region. This is the alcohol, metabolizing gene region, which is a very well-established genomic association in alcohol consumption. But we did identify genetic risk variants that were novel and potentially specific to our populations. And when we increased to close to 500,000, we were able to see additional peaks, and including peaks that were not identified before, reported before. And here in this little map plot, we're showing where our study falls compared to previous large-scale efforts, and we still surpass both by identified variants and sample size. This is a gene-based GWAS analysis, just to give you an idea of the genes that were identified. We did genetic correlations. Genetic correlations were as expected, with traits related to substance use or risk behavior. And we're looking at polygenic risk scores. First, the variants explained of these polygenic risk scores. We see that the variance explained is pretty, as expected, is low, but higher than those reported for Latin American population. And by comparing with different subgeographical groups across the American continent, we see a variation in the variance explained across these different groups. And we compare that to Saunders et al. 2022 study, which is a polygenic risk score generated from an European ancestry GWAS. And we compared the performance of these PRS for multiple subgeographical groups. And we see, for example, for those based in South America and Central America, our GWAS or PRS-derived GWAS-derived PRS in Latin American populations significantly outperforms those that were generated in European ancestry. Here reporting novel loci, well-established loci as well, and we observe varying PRS transferability across different geographical groups in Latin American regions, and we hope to increase sample size further, expand to other psychiatry traits, and also accounting for admixture. If you want to learn more, please reach out to us. This is our website and the QR code that will take you directly there. Thank you very much. And these are just pictures of my lab.
Thank you, Dr. Montalvo-Ortiz, for that presentation. Maybe we can take a few questions from the audience. Does anyone? I will begin, as the audience will. I have two questions. The first about your experience in the consortium. I'm really interested to know what recommendations could you give us to make partnership growth? You share with us an experience that has really grown, and what works for you at the consortium?
Right. So I could talk about membership growth by first across Latin America. I feel like trust is something that it's very important when establishing partnerships and credibility. In order to have a very that establish that partnerships, we have to be very transparent from the very beginning. So things like if we're looking at data access, establishing everything on paper, like data usage agreements, make sure that the partnerships is a two-way direction, that it benefits both parties, and that, especially I'm talking from an institution based in the US, when I'm establishing collaborations in countries in Latin America, I make sure that they also can benefit from the collaboration, and hope that, if they collaborate with other colleagues across, the US region, that they make sure that they can have space to have a leadership role in that collaboration. So in terms of collaboration with people in the US coming from Latin America, I would say that make sure that things are very clear at the very beginning. What are your expectations of the collaboration? And follow all the regulatory processes for any data sharing. Even in grant submissions, you wanted to have explicitly mentioned what your role will be. And if you want to have a bigger role in leading the work, or an opportunity for your lab to have a bigger role in a specific project, you have the right to claim it. And if the partners disagree with that, then they're not being the right partners to do so. So that's why consortium like this that can facilitate those collaborations are needed in order to establish a playing field, like a fair playing field for research groups, regardless of where they are from, or if they have high impact publications or not. The goal of the consortium is to facilitate collaboration, but make sure that the collaboration it's inequitable, could be sustainable long term, and that it can provide a leadership role or an engagement, an active engagements of the researchers in the main study. We know that traditionally, or historically, I have to say, most of the collaborations so far has been researchers based in Latin America, providing samples, and then not even being acknowledged or not being involved in the data analysis or the research study. And I believe that, or at least that was the hope of this consortium, for those researchers to find a platform and a home to make these collaborations, and make sure that these collaborations are done in an equitable manner, that the researchers, if based in high income countries, if they don't follow the initial agreements, that they can be accountable for, and just claim, establishing a platform for researchers in Latin America to claim a bigger role in these studies, that can go beyond just a mere authorship, in the middle of the authorship list, but instead, giving a chance to lead the work as well. So I know there are different partnerships. That's the one that I had in mind, and that's the one that I feel is the greater gap right now. But I'm happy also to talk about other kind of partnerships as well.
Of course, thank you, Dr. Montalvo. Apparently, we don't have much time left for questions, but we will reach to you with more questions from the audience and, of course, from us. Thank you again for your presentations and your experience. Please, again, for Dr. Montalvo.
Thank you so much, and reach out with any question. I'll be happy to answer.
Thank you.
Bye-bye.
Our next speaker is Dr. Kun-Hu, who will discuss circadian rhythm, sleep, and brain health. Dr. Kun-Hu is a professor at Harvard Medical School and a member of the sleep medicine program at Brigham and Women's Hospital. His research focuses on the dynamics of biological rhythms and their relationship to brain health and aging, where he has developed an innovative approach to the measurement and analysis of circadian rhythms. Welcome, Dr. Hu.
Hello, everyone, can you hear me? Just confirm. Testing. Can everyone hear me?
Yes, I can hear you. Can you hear us?
Yes, good.
Perfect.
Should I share my screen or are you gonna load the slides for me?
Can you please share your screen?
Okay.
Thank you.
Just bear with me. Sorry for the delay. Okay. Okay, let me see if it's working.
Yes, we can
Can you
see
see
your
my
screen.
slides?
Yeah, we can see it.
Okay. First, thank you so much for the invitation. Unfortunately, I cannot be there in person. Hopefully next time. So in this talk, I would like to brush through some of the concept in chronobiology and try to convince you the significant importance of circadian rhythm sleep and how that relate to the brain health. In this case, I'm going to just focus more on the dementia or Alzheimer diseases. So first, before I forgot, I would like to thank all the people who have been working with me, and many of them are my mentors or collaborators. So this is the, just by the way, this is some of the people when I work in the Brooklyn Women's Hospital. Just by the way, I already moved to MGH. The end of 2003, I think. So we trying to build a new center here, and then this is some of the group members at this new centers which are gonna launch in July. So I'm gonna skip all those details or introduce each people, but trying to thank all the people who involved in the project. I try to mention the name for each of the project or papers published in next slides. So in this talk, there will be 4 parts. First, I'm going to introduce you to the circadian control system. And even though I put the sleep in my talk, but then I'll focus more about the circadian rhythms. And then, of course, I can explain why. And then, of course, the second the part will be regarding the healthy consequences or effects of circadian rhythm and the misalignment. Then the third part is how that link to the aging or dementia. So hopefully we have time to go through some of the discussion. So as you may all know, when people talk about the circadian rhythms, the first reaction is it's an evolutionary adaptation to Earth's rotation because of sun there. So we do have a 24-hour alternations of environmental factors, including light, temperature, and many other factors. So the first evidence there's such a biological clock is observed in 1729. People observed some plants, they have a day and night pattern. And just curiously, they put the plants in constant dark room, and they still see such a day and night, or should I say 24-hour rhythm. So that's why people starting thinking there's such an internal biological clock. So following that observation, of course, people observe human ourselves and also animals to show some of the also daily rhythm. And in this diagram, we're trying to put some physiological function or output, how that changes across the day. But then, unfortunately, I cannot, give the rhythm. Just give you some of the the peak or trough of the physiological output. And some of them you may argue, this is not so accurate. I'm going to explain the reason why, okay? But anyway, the rhythm are there, and also the most important thing is this rhythm persists under constant environmental conditions, which I'll also show you some of the methods how people observe this rhythm. So actually, a long time ago, this started in 1970, people trying to prove there's such intrinsic rhythm, like in the plants. They actually studying the so-called human circadian studies in the cave, okay, to avoid outside influence. And then, of course, as a scientist, they also moved the human study into a laboratory and try to control all the environmental conditions. Here, I do not want to spend too much time talking about the details, but then the general idea is trying to separate the sleep-wake cycle from the endogenous circadian regulation. So this is two typical human circadian protocols we use. One is called the forced synchrony, another one is called the constant routine. And concept for the forced synchrony trying to make the sleep-wake cycle instead 24 far away from 24-hour. In this case is the 28-hour. You can see the black bars shift 4 hours every day delayed, okay? And the other one is keep the posture or all the conditions constant throughout the day and night so that you do not have a sleep. So that abolish the sleep-wake cycle, trying to see if there's an endogenous circadian clock. Of course, you may argue this is not really happened in the real world, but that is the one way to distinguish whether your body rhythm are due to sleep-wake cycle or due to intrinsic body clock regulation. So I'm not going to go through too much, but that you know, for example, this is a real human signal from core body temperature. And you can see when we measure the core body temperature, how it fluctuates the day and night. And then when you shift the sleep-wake cycle, when you sleep during the day and wake up during nighttime, actually the amplitude of this daily rhythm dumped or suppressed. And then also during the constant routine, when there's no sleep cycle, there's still such a robust rhythm, which means there's an intrinsic regulation. In terms of what controls such intrinsic rhythms, there's many studies. I think the most early study trying to establish what is this neuron circuitry for the circadian control. Actually, I want to mention these two gentlemen, Dr. Robert Moore and Irving Zecker. They show in the SCN, which is suprachiasmatic nucleus located in the hypothalamus, is the master clock in mammalian brain that controls lots of 24-hour rhythm in behavioral physiology. Okay, there's a very specific pathway how the light can get into the eye through such a so-called retinal hypothalamus trait and reach the SCN and modulate all the circadian rhythm of the physiological function or processes. This is another talk if someone interested, but I do not have time to go through with you which of the some of the in addition to the red and corn in the eye, there's another third melanopsin receptor in the eye can help to modulate the light inputs and into the system and modulate all your circadian rhythms. And the evidence showing the SCN is the master clock comes from the several important studies. One of them, of course, trying to show in the SCN showing daily rhythm, or should I say 24-hour circadian rhythm, even during the constant darkness, okay? But this is something which is not for me, this is very important, but there's actually beautiful study in animals showing the SCN mammal is the real circadian clock is from Martin Rotter's beautiful science paper, in which they find animals with mutation and the intrinsic circadian rhythm instead of 24-hour is close to 20-hour. And what he did is he removed the SCN from these two group animals and the implant of them switch the SCN and showing the animal actually they 24-hour or the circadian rhythm the period change switched. So this is the very beautiful work showing in animal model SCN is really the master clock. And of course, I'm not going to talk too about the SCN itself because SCN is very small, about 1 millimeter and by size. But then this is the most intense neuron tissue in the hypothalamus. There's like 30,000 to 50,000 neurons in such a small area. And then there's a lot of people studying the neurons within the SCN to check how they can regulate so many different physiological outputs. Just give you some of the ideas for the SCN in vivo. There's a rhythm remained in constant darkness. In addition, people harvest the SCN. If you do not, destroy the network too much and put in dishes, maintain it very well, and then you can still see beautiful circadian rhythm. Okay. So of course, when we talk about the circadian rhythm, we have to mention the 2017 Nobel Prize shared by 3 gentlemen. They find their mechanism for the circadian regulation, use Drosophila as a model. So this is not my topic today, but very quickly, the circadian regulation involves a transcription, translation, feedback loop, involve lots of genes and proteins, and then they have such kind of like, for example, transcript the diaper from the parent team. This is in Drosophila. It's more complicated than in mammal, but involves the BIMA and the clock. Okay, so if you're interested, you can read a little bit more work. And then after the discovery of the underlying molecular mechanism of the circadian control, people starting to notice actually not only within the SCN, but there's a lot of different peripheral organ and also some of the brain tissues. They have such same molecular circadian mechanism. So it's within lots of cells instead of only some of the SCN neurons. And this is make the picture very complicated, which means the SCN receive some of the light information, but they talk to different so-called peripheral clocks. So there's a network here to together to regulate many, many different physiological or biological processes, and including one of them is including sleep. Okay. Unfortunately, I do not have time to go through all the details, but I'm going to cover more physiological level, some of the process in my talk. And if you're interested, you can read some of the review articles. So I'll move quickly to the next part. In this part, I focus on some of the circadian influence on physiological functions. Of course, the first one I like to talk is sleep. So instead of a central around sleep, if you want to know how the sleep linked to brain health and many other, physiological consequences and well-being or economic, burdens. So I think Dr. Diego Gallenbach gave a very nice review recently, and I'm very happy to get involved in this review article. You can look at it a little more carefully, okay? So first, when we talk about sleep, one question is very simple. Why do we sleep? And then, of course, many people argue if we do not need to sleep, we can work longer. So I think you may already know the answer is not so simple. So why do we sleep? It's a very difficult question to answer. There's a different theory. I listed some of them here, and you can check more online. And none of them are perfect, but there are some of the evidence supporting them. Not always, necessarily cover all the things about sleep. So, for example, one thing you may think about very from biological understanding is energy conservation, right? We use the energy during the day to get something, but we need to reserve the energy during night, okay? However, this does not explain many things we know about sleep, okay? So, for example, sleep is not just resting. So you may see online, some people said, I'm not sleeping, I'm just resting my eye. Actually, this is not true. Sleep is not just resting, okay? There's lots of evidence supporting that. First, sleep is not one state. There's a different process and different active brain regions during sleep. And then one thing you may want to know is there's like right now, there's different sleep stages. For example, when you get into the bed, you're starting awake, and then you goes to light sleep stages. And then you slowly go into deep sleep stages. And then you're not stay there forever. The so-called 90 minutes cycle. In other words, they get back to the light sleep and then goes to another sleep stage called rapid eye movement. In these sleep stages, most of the dream happen, okay? And this is around the 90-minute cycle is not so kind of constant throughout the whole night. And you can see from this cartoon, at the second half of night, the duration of the REM sleep getting longer. So that's why you have your dreams most of the time during the second half of the night. More than that, there's many studies showing in sleep stages, such as dynamic changes relate to the memory consolidation. This is another topic, but when we say memory consolidation, give you an example. If you, during the day, you learn something, for example, in this slide, you learn about the sleep stages. And then you digest such information, actually during sleep, if they have a good, healthy sleep, during sleep, such a learning process can be consolidated. So you your memory can be repeated during the sleep. So I'm not gonna talk too much about it, but if you're interested, you can check again, see if memory consolidation, you can find lots of literature. Recently, people even saying, we can edit your memory in sleep. So this is the same thing like you see the movie, what's that movie? Inception and many other movies showing how can you manipulate people's memory during sleep. So as I said, I'll focus a little bit more about the circadian regulation. So actually the sleep regulation, there's actually two processes. One is called sleep drive. For example, I keep awake for a while and then my sleep drive will build up, right? Okay. And then if I do not sleep, the sleep drive will increase. That happened during the sleep deprivation. But sleep drive is not the only things affect your sleepiness or sleep drive. There's another process called the circadian process. In other words, the circadian system sends a signal to make you feel more sleepy at a certain time, but less sleepy during other time. And this is some of the cartoon picture showing. For example, when you wake up enough, you starting feel sleep, and then if you decide go to sleep, your sleep drive will gonna decrease. However, your sleep, circadian modulated sleep, drive will not necessarily goes up. By the way, this is upside down because they're trying to use the circadian trying to motivate your sleep. So that's why people can sleep longer than 8 hours, not just because you need 8 hours for the sleep drive, but due to the circadian modulation. Actually, for the small animals, they never sleep continuous like human for 8 hours, such a long time. So I think this is a very accepted two-process model for sleep regulation, okay? Just give you another example for the sleep influence on sleep. If you let people to sleep at different time of day, of course, they control for many other factors, including the sleep deprivation, other things, and people can easily get into sleep during their habitual biological night instead of during the biological day. And then this is can be shown by the sleep efficiency and the latency. So the take-home message is it's difficult to sleep during the late afternoon and early evening. So this is because the circadian influence at that time, the body is not prepared to sleep. And how the circadian system, regulates sleep. There's many different pathways and hormonal, right? And here I give one of the example is called melatonin, and people know about that. Melatonin is, controlled by, secreted by the pineal gland, but it's regulated by the SCN, okay? And actually, if you monitor the level of melatonin in body, very interesting, people know about this. Actually, during the day, the melatonin level is very low, but then before you sleep, actually melatonin level starting increase. So we call it the melatonin onset, okay? And then after you fall asleep, the melatonin reaches the highest level and maintain there. But before you wake up, actually melatonin decreases. And even before you wake up, decreases some level. Okay? So I want to give you the impression is the body actually trying to prepare you to get into sleep or wake up. So this is something, as I mentioned at the beginning, the circadian rhythm is to prepare you to as adapted to the sleep-wake cycle or environmental changes. Okay. So of course, one of the things you may think about it in terms of if you disrupt light, what will happen? And one of the very known phenomena is called the seasonal effect. Actually, due to the different light exposure during the day, how that affect your melatonin, so make your changes sleep-wake cycle. And also, one of the things you may notice is if during the nighttime you have the sudden light exposure, they're going to suppress your melatonin. And then there's two effects. One is acute effects. The light during night wake you up, right? But then actually there's something called the circadian, phase shifting. In other words, they delay your or advance your body clock based on where or when, the light exposure happened. So in other words, if you have your light exposure before your sleep time, usually they're going to delay your body clock. But if you have light exposure just before you wake up, some circadian phase, then they advance your body clock. So that's why one thing you may heard from lots of chronobiologists tells you, don't expose a bright light before you sleep. Otherwise, you cannot get into sleep because they suppress the melatonin and delay your clock. And then, of course, you also know during morning, if you have a light, it can help you wake up. Okay. Since we talked about this circadian regulation, and then one concept that people always get confused is about the when I say circadian rhythm of around 24 hours, why is that? Okay, so actually, if you let the constant darkness, you measure the circadian period. It's not necessarily exact 24 hours. And sometimes it's less than, sometimes more than. This is like a famous Dr. Ashcroft, the founder of the chronobiology, and our Charles Czeisler. They found that there's a distribution. Okay, not everybody have exactly 24 hours. And you may ask, this is tiny changes, right? 23.9 or 8 to 24.6. What the work on this one make you think this is important? Actually, surprisingly, if you check the intrinsic circadian period, that is related to lots of other things, your melatonin onset time and relative to your bedtime, okay? So this is some of the very important physiological properties. So sudden change there can affect dramatically timing of your behavior or physiology. If you don't think this is very easy to understand, I'll give you one specific examples. So for example, people understand some of us trying to get up earlier, sleep earlier, okay? And then physiologically, we can find the core body temperature can drop earlier or before the sleep and melatonin level increase earlier. So these people usually have a shorter circadian period. So we call it the morning type or early birds. And for me, I'm night owl. And actually, I try to stay awake and wake up late. So, and then actually, my body clock actually should be, have a longer intrinsic circadian period. Okay. So I'm going to come back to this question maybe at the end of the discussion if it's, we have enough time. But then, as this is just an example, so there's lots of, massive physiological difference in terms of timing for different chronotype. And interestingly, Dr. Jean Duffy studied, they find the sex difference in intrinsic circadian period, and the men usually have a longer intrinsic circadian period. Okay, and this is very interesting because that means more men tend to be night owl, which seems like this case based on the epidemiological studies. So I'm going to stop here because this is regarding how the circadian system regulates sleep, how they make the sleep cycle difference based on the intrinsic circadian system and the properties. So next, I want to go through some of the examples tells you the circadian system affect many other physiological function. Here, I use some of the cardiovascular function as examples. Actually, when I got interested in the circadian physiologies, because my mentor for my postdoc training, Stephen Shea, he asked me the question, why the heart attack happened more in the morning hour between 9 to 11? Is that just due to your wake up? You're not prepared, or there's some of the circadian regulation make your system more vulnerable at that time. And I'm so happy. This is my PhD thesis, the major article showing actually using such a in laboratory controlled environment, which showing there's endogenous changes in the cardiac risk. So, and then this is leading to a field advance trying to answer the question whether the circadian system involves lots of cardiovascular, events. And actually, we do repeat many other instances like the stroke, morning stroke or syncope, many other things. They do relate to the circadian regulation. I'm not going to spend the time to go through all the details. For example, in this study, we're showing the tilting change the position, and then that maybe introduce more high syncope during the early morning hour. But the peak actually is during nighttime. However, we we do not wake up during nighttime, have such tilting or posture changes. So, and also we use the in that laboratory study control other sleep cycle, other factors, we're showing the circadian system also modulate many physiological output, including the blood pressure, heart rate, autonomic function, and we showed how even not only affect the baseline system, but also the response to some of the stress. Here's the tilting, but also to the exercise, many other things. I'm not going to go through all of them. So when people at the beginning use the laboratory study trying to focus on the endogenous circadian clock, but in the meantime, when you shift the sleep work cycle, you also create another thing people are familiar with, which is called shift worker. For the shift worker, people trying to work during nighttime, but sleep during daytime, which is exactly happening for some of the period in the laboratory when we set up the the human circadian studies. And one of the most striking factor is the night shift worker is linked to many advanced health consequences, including risk for heart disease, heart attack, and obesity, and diabetes, and depression, and many other things. So that's why our early studies focus on cardiovascular functions. And one thing, I have to mention this one, is the so-called circadian misalignment, or when you sleep during the day, wake up during nighttime, then this circadian misalignment, we can see so-called suppressed leptin. Okay, if you don't know what the leptin do, is lower leptin level make you feel after eating less satisfied, and then triggers you eat more. So this can lead to other things like obesity. Okay. And they use the same laboratory study was showing such changes in leptin, not only the leptin, but also the glucose, intolerance. And actually, some of the people, when they during this misalignment, the glucose tolerance is more like even pre-diabetes or even diabetes. Okay, so this is a huge effect showing the circadian misalignment on the cardiovascular or metabolism. And to simulate the shift worker, my co-mentor during my postdoc training, Dr. Frank here, also used the simulate night shift work. So they check what really happened. Okay, so this is actually one thing we call the circadian misalignment. But what's the misalignment? So people focus on behavior, but actually there's a so-called misalignment of the physiological signals. In this case, you can see very clearly, melatonin usually, as I showed you before, should be higher during the sleep, right? But then when you have a shift worker, then the melatonin is still during daytime. Why? Because the circadian system cannot respond to the environment changes quickly. They need a time. This is just like you have a time lag, travel to different time zone. It takes time for you to change your or shift your clock time. But for the shift worker, in most of the cases, it's a very quick change. And then this is going to generate such a so-called misalignment. Okay. Okay, and then I'm going to skip all the details, but using the laboratory protocol, Frank, and I'm happy involved in some of the studies showing all those circadian misalignment can introduce to reduce the nighttime blood pressure dipping, and also increase the inflammation, and also decrease the glucose tolerance. So I'm not going to repeat again, but if you're interested, we can chat more. Of course, since we talk about the brain health, we want to check the cognition. And then this is well known. There's a daily rhythm in cognition. And the people at the beginning just using the sleep drive model, basically two process to drive you sleep or sleepiness or not. That sleep drive and the circadian drive make the such a kind of model for the cognition alertness. However, as the long time ago people know, this is too simplified. There's many different things you need to consider the daily rhythm of the cognition based on what kind of things you do and the complexity or duration, many other things, aging, or many other conditions may affect the such a daily rhythm of cognition. This is also some of the thing called sleep inertia. If you don't know, that means when you wake up after sleep, it takes a while for you to recover in terms of cognitive performance. In other words, at the beginning of wake up, then you have some so-called transient impairment in cognitive performance. So actually, using the same in laboratory study controlled sleep cycle, we show at different circadian time, not your sleep wake decide, but your circadian time, there's an endogenous circadian rhythm in such a sleep inertia, okay? And here, this biomarker shows the sleep inertia is higher at the biological nighttime, okay? So in other words, you wake up here, the sleep inertia is larger, and then you take a longer time for you to recover from this transient impairment in cognition.
Excuse me, Dr.
So-
Hu?
Yes?
I'm sorry, we have just a couple minutes. You can finish like a bit faster, please.
I cannot hear. Your voice cut a little bit.
We have a couple minutes more to finish your presentation.
Yes, I
Please.
will.
Thank you.
Yeah. I thought it's 40 minutes, is that correct?
3 to 4? 3 minutes or so.
Okay.
Thank you.
I'll speak up, okay?
Thank
should
you.
be
Thank
easy.
you.
So of course, we also check the cognitive performance during the circadian alignment, showing that's true when the circadian alignment, the performance is worse. Okay. So I'm gonna skip this and jump to the last part, which is related to how that linked to the brain health and the dementia. Okay. So according to such 2021, this is from NIH, they encourage people to study how the sleep circadian rhythm and how that linked to the dementia pathology. Okay. So that's why in last almost, for me, I think last 12 years, my research focused on dementia. Okay. And one of the things you may understand in why people believe the circadian misalignment relate to the dementia is also come from the shift worker. Shift worker not only have the high risk for other vascular events, but also for cognitive decline. So that's why one of the hypotheses is from the shift worker, that is because of introduced the circadian sleep disruption may lead into some of the impaired brain function acutely, but also chronic or in long term. So I studied such a field, try to look at some of the other different circadian biomarkers, not just rhythm, but also some of the fractal. This is another talk. I'm not going to spend time on it. But anyway, so we do find such like changes, how this circadian regulation degraded with aging and in Alzheimer's disease. Initially, it's cross-sectional study. And then we also showing how those circadian physiological measure output linked to the SCN neuron transmitter within the SCN. Okay. Using longitudinal design, we also to showing how the circadian parameters, including the amplitude, phase, and also many other fractal measures, how that changes with aging, and how this changing accelerated after the onset of Alzheimer's disease. In addition, we also showing at baseline, if people with more degraded circadian regulation, then they have a higher risk for develop Alzheimer's dementia. So this is a kind of evidence for bidirectional relationship between the circadian disruption and the development of dementia. So one thing also, one of the other type of the circadian disruption people know about is called daytime napping. We also showing consistently increased daytime napping in older people seems to link to the risk for dementia. Okay. So I jumped through. The reason is I want to save at least 1 minute or two for this discussion quickly and summarize. There's such a progression of the Alzheimer's disease and the related dementia, and the circadian control network, its dysfunction play a roles at early stage and maybe accelerated such a progression. So many things, we have a review paper showing what's the potential pathways. Due to limited time, I cannot go through them. Hopefully I convince you at least one of the things is cardiovascular link. And actually, one of the limitation in all previous studies is not too much in Latinos. That's why I'm showing some of the examples here, why what we know from the white people in the US may not apply to Latinos. And that's why we have such ongoing study with the red light, trying to understand better. It's the same thing happened in Latino American countries. So there's a few projects I want to highlight. One is the blood pressure. I want to share with if August, other people here. It's got scored, but not good enough. And currently, we're trying to review the summary statement, the comments. Hopefully, we can submit in July. Okay, I believe it is addressable. All the comments. I'm going to share the more details later. And the other grant we just resubmitted a few months ago, and they're going to be reviewed soon. So all these two grants are focused on cardiovascular dysfunction and see if that can be the linking the circadian or sleep disruption to brain health in Latin American countries. Okay, and the other thing is about sleep. Of course, as I did not focus too much on the sleep, but sleep is have different sleep stages, and the sleep EEG is one of the best physiological output can be used to maybe predict the future risk for dementia. Okay. So Shahab has ongoing K99. Actually, Sebastian have just submitted a K99 with me, Agas, and Dr. Miller as the core mentors to study the high order interaction in the EEG and how that link to the future risk for cognitive decline. So, and also the good news, we just got approval for submit a huge R1, which is greater than, I forgot exactly, maybe $15 million. So we're going to use the study the sleep EEG red light participant. Okay, so I'm going to talk to people more. There's many other things I want to discuss, but without enough time, I want to go to the last slides. Honestly, all the things I told you today mostly focus on the people in laboratory. The picture about circadian disruption in the real world is still missing because many things can affect such a rhythm and then alignment, including light, temperature, humidity, meal, and drinks, timing, and amount, and also many other things. So we think this is very important to understand which factors can contribute to the circadian disruption in the real world should be the first step. And also based on the real longitudinal study, if we can find out the optimal alignment between this rhythm and link to the better cognitive outcome, that'll be the first step before we can use the circadian therapy to maintain better circadian health and to prevent or slow down the development of the dementia. So I'm going to stop here and thank you so much for your time.
Thank you, Dr. Hu, for that presentation and that final part where you discuss all these topics interconnected. We have some questions from the audience. We have time for one, maybe two questions. I think that there's not from our part. I was very interested in that last part of you when you show us that map with all these topics that we have to take account when we engage in trying to get better sleep. Can you give us maybe some examples of that things that we can take in considerations from our better lives so we can reduce that risk in our health through the lack of sleep or bad habits in the sleep?
Yeah, so I think sleep is one of the component here, sleep cycle, but I want to emphasize this. In many of the results I showed you, sleep deprivation is cannot explain those adverse consequences. It's a misalignment. So I'm not saying sleep is not important. It is important, yes. But then sleep itself can create such alignment in the body clock. So why those light and other external stimulus is important is because this we call it the Zeitgeber, which is circadian stimuli, can help you to maintain the circadian clock. One of the example I mentioned is about the light exposure. If you have a regular light dark cycle, then actually your circadian system is more stable. Actually, that's how I help us to shift it to the different time zones. Okay. So if during the daily living, you have an irregular light exposure, especially during the nighttime, you have light exposure to mess up your clock, that'll be bad. And then that clock will in turn to affect your sleep quality and many other things. So this is a vicious cycle. introduced by some of the external stimuli, and then which can amplified by such a circadian misalignment process. So I think this is all the thing have been showing is quite relevant in laboratory study, but we really don't know how much in the real world. So that's why I think there's other thing I want to emphasize again is the chronotype and the genetics. Because of circadians, lots of gene protein that involved, and I did not even focus on the epigenetics. That's another big topic at this moment. So in other words, how do we understand those external conditions affect the alignment between different circadian rhythm? How that is mediated the genetic effect? This is a huge question. I believe we need to some of the large field study with multidimensional physiological monitoring and including other external factors. So thank you.
Thank you, Dr. Hu. We'll be remain attentive to all the future work you share with us. Thank you again for your time.
Thank
Please
you.
give it again
Yes.
for Dr. Hu.
Yeah, thank you.
With this presentation, we end this part. We'll be in a short break. We will continue at 3:45. 15 minutes for a coffee break.
So I'm going just to introduce briefly Dr. Rafi Haddad. Dr. Rafi Haddad received his medical training from the University of Messina in Italy and completed his residency in neurology at Rambam Healthcare Campus in Haifa, Israel. He's an Atlantic Fellow for Equity in Brain Health at the University of California, San Francisco, and serves as a neurologist at a clinical fellow at the Stroke and Cognition Clinic and the Movement Disorders Clinic and Rambam Healthcare Campus. So we're going to start now with the presentation of Dr. Haddad.
Ladies and gentlemen, we are here with Dr. Rafi Haddad, who's going to deliver the presentation, Brain Health in Underserved Populations. He's a cognitive neurologist, works at the Stroke and Cognition Institute at Rambam Healthcare Campus in Haifa, Israel, and he's Senior Atlantic Fellow for Equity in Brain Health, Global Brain Health Institute, the Department of Neurology in the University of California, San Francisco, UCSF. Dr. Rafi Haddad, welcome, and we are all ears for you. Thank you very much for your presentation.
Thank you so much, and hi everyone. So today we are going to talk about brain health in underserved population. So for the beginning, I want to explain what is brain health. So brain health is our ability to keep and to preserve our cognitive abilities, which will let us function very well in our life and navigate daily life effectively and realize our potential in the life. So there are many factors that can influence brain health, including our physical health, learning, social connection, and quality of life. And what we mean when we say underserved population, who have limited access to healthcare services such as neurology or memory clinics. And there are a lot of barriers, and between these barriers are geographic locations, race, ethnicity, some beliefs of some populations, and stigma, trust, poverty, and insurance status in each country and in each place. So these populations are also underrepresented in research and in clinical trials. Why this concept is important? So as we know, the lifetime risk of dementia after age 55 is 42%. And this risk is higher in Black adults, and it arrives to 45 to 60% of the population. In 2019, 57 million people had dementia worldwide. And this number is projected to rise to 153 million by 2050. And in 2020, each year it was diagnosed about 500,000 new cases of dementia. And this number is also projected to rise to 1 million in 2060. So why brain health matters in low middle income countries? As we know also, 2/3 of people living with dementia reside in low and middle income countries. And the rates of undiagnosis of these disorders are high, and it arrived to about 90% of cases are undiagnosed. There's a higher prevalence of vascular cognitive impairment, low availability of biomarkers, and also lack of appropriate neuropsychological tools for the identification of these diseases. So it's clear that there is an urgent need for policies that enhance health aging with focus in health equity and underserved populations. According to the Global Burden of Disease study that was published 1 year ago, in the world, there was about 443 million cases of disability that is related to neurological disorders, and which make neurological condition as the #1 leading cause of disease burden worldwide. And if we look in this table, we can see stroke, we can see migraine, we can see dementia, we can see meningitis and epilepsy. All these diseases are neurological diseases that are affecting our brain health. And if we look what's happened in low middle income countries, the prevalence of these conditions is higher than other countries. And here also we can see that also there are psychiatric diseases that are more prevalent, and the drug and alcohol disorders that are also more prevalent in low middle income countries, which can explain for us that also in these regions where we have higher risk of cognitive impairment, also we have more conditions affecting our brain health. And with all of this, we know that we have missing tools for diagnosing and treating, and we have an awareness about prevention of these disorders. So what are the determinants of brain health? Brain health is influenced by many factors, including age, sex, physical health, environment, and we cannot forget conflict and safety and security, learning and social connection, access to quality healthcare services, and also in low-middle income countries, we know that infectious disease is one reason, and socio-economical disparities are also considered. And in a work that was published by Dr. Santa Maria, we can see that in Latin America, social determinants of health, lifestyle work, cardiometabolic risk factors, and mental health symptoms influence our brain health more than age and sex. And these patterns were more pronounced in low-income to middle-income Latin American countries. So the strategy, what should be? The strategy is prevention. Prevention is the key for keeping our brain health. So there are two types of prevention that generally we talk about, primary prevention and secondary prevention. When we talk about primary prevention, we are talking about treating the reasons before disease onset. So we need to focus on all the modifiable risk factors that we know about, such as hypertension, diabetes, obesity, physical inactivity, smoking, diet, and etc. And also in secondary prevention, the importance here is to early identify and intervene to prevent the progression of these disorders. So in the Lancet Commission, they published also 1 year ago that about 45% of dementia risk factors are preventable. And if we can look on this table that we have here, we see that modifiable risk factors, they can start from early life to late life, including education, including vascular risk factors, smoking, traumatic brain injury, depression, and also some social conditions in later life and air pollution. And so we can see that also if we have more prevalent cognitive disorders or more prevalent risk factors, but these are the factors that we can work on and we can change so we can prevent any damage to our brains and keep brain health. So when we address these factors, we decrease vascular damage, we reduce dementia neuropathology, we reduce stress and inflammation, we will build cognitive and brain reserves, which will prevent and delay any dementia. So now I am talking from my point, from the world where I work. So I work in Israel. I work here in the north of Israel, in Haifa. And if we can see, I put here where we have cognitive clinics in Israel. So in Israel, we have mainly the cognitive clinics or in the north in Haifa, or there are another two cognitive clinics in the region of Tel Aviv. And if we are talking about how many people, what is the population of Israel? As you see in this picture, the population in Israel is super diverse. Also, if it's a small country, but we have people from all over around the world. We have mainly Arabs and Jewish in the country. And as I am working in this country, and I am the only Arab cognitive neurologist in the country, so when I came to work in this field, I saw all the disparities that we have, and I decided to start to make change. And if we look in Israel, how it's composed, so about 21% of the country is composed from Arab population, and the Arab population have higher prevalence rates of dementia, have higher rates of illiteracy, and more people with education less than 12 years, and they have more consanguineous marriages, and they have lower socioeconomic levels. All these factors, as we said before, they are influencing brain health. So we can see already the disparity here and in this point. And also we know that Arabs have more cerebrovascular risk factor, more ischemic heart disease, and mortality from these diseases. And we know, according to all studies that were performed here in Israel, a lot of studies were saying that Arab people have more cognitive impairment than Jewish. So it's clear the disparity and that that the Arabs are underserved population. And we can say another word that was published before I started working in the cognitive clinic in the north of Israel. And we can see that the Arabs were underrepresented in our cognitive clinic. And we don't need to forget that there was no one who can read them because no one was talking their language. And also we know that when we don't talk with people in their language in these countries, we cannot communicate with them and they cannot build trust with us, which may prevent, they will not come to visit us in the clinics, and that's why we see this number. And then when they came to the clinics at that time, they came in more advanced stages, and they were younger in age, but they have lower mini-mental scores than Jewish, and they have higher rates of vascular dementia. So is it clear that this is underserved population? I think we said why they are underserved population. So only one Arabic-speaking cognitive neurologist in the country, lack of culturally appropriate cognitive assessment tool in Arabic, lack of normative data in Arabic, and they are underrepresented as it's expected in clinical trials. So the clinical challenges that we are facing and we faced here in these years, first of all, late referrals, limited awareness of early signs of dementia. And this limited awareness is not only among patients, but also among caregivers and among also healthcare providers. And we know that cultural interpretation of some symptoms is also a challenge because like some people think that in older ages, it's normally to forget. And that is something normal of aging. And this is why they don't go to check what they have. And they have also limited access to cognitive clinics. And also for now, assessments are costly and they are not covered by healthcare baskets. So sometimes people need to pay from their pockets to these cognitive assessments. So what is the strategy and what we are doing for now? So first of all, we need to change what we have. As I said before, we don't have normative data for our population. We don't have cognitive assessment tools for our population. So a work that we are doing with the UCSF and GBHI, with Dr. Elena Tsoy and Victor Valcor, we are working on introducing cognitive assessment tools to Israel. So there is the harmonized multi-cognitive assessment in diverse population battery that we already culturally and linguistically adapted to Arabic. And this battery is including digital tools and the traditional tests that were used before. And also, we are now enrolling patients to collect normative data for cognitive testing. And this will be the only battery in Arabic that was validated and culturally adapted for the population in Israel. And I think that it will be the only one in the Middle East that can help anyone talking Arabic. And another point that is super important in addressing these disparities that we have, as I say, there is a lot of unawareness. So if we don't work in ourselves to increase awareness, we cannot help this population. So it's super important to have education through community centers and schools. And we are working on training local healthcare providers to understand cognitive symptoms. And I think these small steps that we are doing are a key for making a change and changing the future that we have here. So our hope in the future to have more grants for capacity building, to teach more people, to include more people also in research, also in clinical activities, to be able to address all the disparities that we have. Also, if not possible all, but at least to make some change. We need to expand this Arabic language cognitive research and biobanking, not only in Israel. I think it should be done for all the Middle East where Arabic speaking population doesn't have cognitive assessment tools that are valid and culturally appropriate. So we hope also to use these digital tools and the tools that we are having, and maybe also AI for screening some remote areas where not always we can arrive and where people cannot always come to our clinics. And impact cannot happen if we don't have a collaboration also with people who have more experience and also with people who have the same problems from underserved regions. And all of this cannot happen without any resources. And we all know what is happening now in the world with all the struggles in bringing resources to help underserved populations. So we need global efforts. We need to invest in inclusive regions, to build trust in underserved population, to train culturally competent clinicians, and to advocate for brain health equity. And I think this is the super important actions that we can, each one of us, work on. And I think everyone who is listening to this talk have some underserved population that or he treat or he know about. So we need to help them to get access to medicine and to get access to cognitive neurology and to get access to the possibility to prevent cognitive disorders. So in conclusion, brain health is a human right. Equity in diagnosis, care, and research is something that's possible that we can do. We can prevent neurological disorders and improve brain health. So why we are not doing that? There is a need for global commitment to brain health, particularly in underserved population. We need to advocate for policies that promote equitable access to brain health services. And we don't need to forget that brain health leads to positive social and economic impacts, contributing to more productive society. So thank you all for listening to this talk, and hope that it's giving us a little bit of hope and thoughts to what we can do in the future. Thank you, everyone.
Thank you, Dr. Haddad, for your very interesting presentation. I would like to ask you two questions that I think
mission that we have for our population to go and advocate and explain what to go and to talk to the people. These are the people who need us. And if I will stay in my hospital or in my office and I will wait for them to pay to come to visit me, they will not come never. So I think this is a mission that we have for our population to go and advocate and explain why is this concept important. Because like most of these cases, we can really prevent. And what I see in my experience here, we see a lot of people that come like with a lot of vascular cognitive impairment. And like these people, never they treated their cholesterol, never they treated their diabetes. And just when we found that they are cognitively impaired, we started searching that. So also all the medical conditions are not treated. The healthcare system is not enough good to take care of these people. So I think it's our responsibility to go and to advocate for them and to explain for them the importance of what we are doing.
Thank you very much. And the other question is like, what are the two bigger barriers you find like doing your work right now? And what are the two biggest opportunities you identify in there?
Can you repeat the question, Laura?
Yeah, what do you think about like what are the biggest opportunities you identify at your work, at your current work, like right now, and what are the two bigger barriers you find in your current position, like for prevention specifically and promoting brain health?
I am not sure that I understood the question, but I think that what you are asking about is like, what is the challenges that we have now?
Yes, certainly.
Okay. So the challenges that we have is like that, as I said, cognitive clinics, we have like few people working on that. Like in all the country, we have about maybe 10 people who work in cognitive neurology, Arabic speaker, only one. So the problem is to have more people working on that. And also to train a lot of healthcare providers how to identify cases when they start and not let them stay like when they are advanced and to send them to us, to our clinics. Like some of also like primary healthcare providers, they think that they should send the patients for a control or a cognitive neurology clinic just when they have functional impairment. And this is super wrong. We need also to advocate like not only for people to reach out to them and to explain why this is important, but also super important is to train healthcare providers to early identify any problem and to explain to them what is the importance of our work and why all this prevention is important as primary prevention and not secondary prevention.
Thank you very much. And what about the opportunities, like the good things you find right now? Like you have a good reception on the professionals that are receiving your training and about the community that is like just getting more involved. How do you think like this?
Yeah, you know, we say something, you cannot clap with one hand, you need two hands. So also I cannot do all of this work by myself. So, when we work as a team. So we work also neuropsychologists, we have like a social worker, and we have other physicians who like also if they don't talk, we can like sometimes work together and we do all what is needed together. So the importance here is really to involve more people in doing that and to involve more the community and more healthcare providers, because when we are doing something, we are not doing like just for them. We need to ask them what they want. So it's important also to listen for also the healthcare provider, what are their problems? So what they need, what they want to learn about, and also the patients, what they don't know and what we can explain to them. And then we can go and to talk to them, because it cannot be only one way. You need all together. Also, the care providers need to be involved. We need to be involved. Other people in the team also, if they are not physicians, they need to be involved. So it should be a work team to address all these problems around.
Thank you very much for your answers, Doctor. I think it would be it was a great pleasure for all of us having you. So just thank you very much, and it's an honor to have you here in the Congress. Thank you very much.
Thank you so much.
At the distance, thank you, Dr. Haddad, for your time. Thank you, Laura, for the questions and for recording the presentation. If you have questions, of course, feel free to engage in a conversation with Dr. Haddad. Next, we have a live session with Dr. Nilton Custodio. Dr. Custodio, can you hear me?
Yes, I hear.
Perfect, we can hear you as well. And I'm afraid we can't see you. But we can see your presentation. That's perfect. We can see you now.
Okay.
So Dr. Nilton Custodio will be talking about initiatives to modify risk factors for dementia in Latin America. Dr. Custodio is the medical director of the Peruvian Institute of Neuroscience and principal investigator of the research unit at Clínica Internacional in Lima. He is a neurologist with a master's in medicines, a master's in neuroscience, and a PhD in neuroscience from the National University of San Marcos. He is a fellow of the American Academy of Neurology and visiting professor at the University Peruana Cayetano Heredia. He currently leads the Red Lab project in Peru, part of a multiple partner consortium aimed at expanding dementia research across Latin America and the Caribbean. Dr. Custodio, the floor is now yours.
Thanks. Thanks for organizers, primer Congreso Colombiano de Salud Cerebral, who invite me to explain these initiatives to modify risk factors for dementia in Latin America. Based on large-scale review of observational studies, the Lancet Commission estimated that approximately 40% of cases of clinical dementia could be attributed to 14 key modificable dementia risk factors. This support that dementia has a multifactorial etiology and raise the possibility that many cases of dementia could be prevented by strategies that act to reduce these modificable dementia risk factors. I will discuss the diverse risk factors to dementia in Latin America, how possible brain mechanisms for enhancing or maintaining cognitive reserve and risk reduction of potentially modifiable dementia risk factors. Then I will explain the scarce non-pharmacological intervention in Latin America, particularly the Latin fingers. Finally, some intends to adapting intervention for dementia to communities for community health workers in Peru and describe the barriers and enablers to dementia risk reduction. In 2024, the Lancet Commission on Dementia did the new compelling evidence that untreated vision loss and high LDL cholesterol are risk factors for dementia, and moved to 4 risk factors from late life to mid life, basically physical inactivity, smoking, diabetes, and depression. And using data from all 37,000 participants aged 45 years or older from the HANG study, the analysis identified 5 principal components explaining 54% of the total variances between the 14 risk factors, indicating a substantial overlap in risk factor prevalence. In the table, you can see relative risk factor prevalence, communality, and weighted population attributable fraction, and weighted population attributable fraction by every risk factors, and calculate or estimate the path for all 14 risk factors was 45%. With data of the phase report in 2017 of Lancet Commission on Dementia, the path for risk factors in dementia in Latin America was more higher comparing with other middle income countries such as India and China. We have to see the potentially modifiable is 56%. And obesity and low education are higher comparing with other countries. Recently, Professor Paradela from Brazil showed the higher path in Latin America, basically Argentina, Brazil, Bolivia, Chile, Honduras, Mexico, and Peru. The key risk factor with large path were obesity, physical inactivity, and depression. And worldwide estimation is 40% comparing with 54% in Latin America. In this way, the contribution of each risk factor across 7 Latin American countries, obesity, physical inactivity, depression, education attainment, and smoking were higher path estimated. Principal obesity in Mexico, physical inactivity in Bolivia, depression in Mexico, education attainment in Bolivia, y smoking in Honduras. Interesting, the path were more higher in Chile and Argentina, and suggesting higher potential for prevention measures. To understand the path calculated by the random effects meta-analysis across 7 Latin American countries, you need to see the early life is represented in green, mid-life in light blue, and later life in purple. And you can see that the weighted path were calculated to every country. Diamonds represent the summary measure, and comparison the reported measurement for diamonds is ideal the global population is shown next to the globes. The overall proportion of dementia cases could be potentially avoided by limiting the 12 modified risk factors for dementia in Latin America was 54%. Obesity, physical inactivity, and depression were the three most important factors in Latin America. In the past calculated by the random effect meta-analysis were higher than the worldwide estimations. Also, the life course model of dementia risk proposed by the Latin Commission report suggests that some modified risk factors on dementia have the greatest influence on dementia risk in later life. It's possible that for some individuals, this may be too late for dementia risk reduction to be achieved. So the probability to include other factors in early life is ideal. This proposed for Professor Bransby classified in non-modifiable, including older age, about E4 and female sex, modified into tests with intervention at social or community level, and modifiable with intervention at individual level. In early life, similar to Lancet Commission, traumatic brain injury, air pollution, low education attainment, but they suggest include low status, socioeconomic status and low occupational complexity. Additionally, modified with intervention at individual level include the other factors, maybe anxiety symptoms, psychological stress, or alterations on duration of sleep. Professor Francesca Farina proposed diminishes the age to intervention. It is meanwhile include modify of risk factors more younger is ideal. So they propose initially the modified risk factors in young adulthood, basically 18 to 39 years. It's important that this population will be modified and diminish probably to risk of dementia. How is possible to diminish, prevent, and delay dementia? Is addressing risk factors could be decrease vascular damage, reduce dementia neuropathology, reduce stress and inflammation, build cognitive and brain research. This approach, evidence-based policies, could significantly reduce dementia incidence. The idea is that multi-components is better than individual components. For example, effects of exercise on cognition have a slow or lower incidence on risk factors in dementia, but include a small sample size, low frequency, and intervention durations. About the diet, is better balanced diet than nutrients. Single nutrients is beneficial for brain health. And diverse clinical trials express the slight possibility to reduce progression to NCI or dementia, but new data is a small sample size, short follow-up duration, regional variations, marginal effect with Mediterranean diets. similar result, but cognitive intervention without other intervention to great studies such as active and hands. Active in USA's, the result have effect on intervention 2 and 5 years. Cognitive trials didn't affect rates of incident dementia after 5 years of follow-up, and similar results with GHANS. Moderate improvement in attention, a small to medium improvement in executive functions such as trimacal tests, symbol digitals, and stroke work, but no change on COAP and DBT. So, multi-domain intervention trial for the prevention of cognitive decline and dementia is ideal. The pivot study, the FINGER evaluate 1,260 persons at risk of dementia based in KID, and main results include significant intervention benefit on cognition, but not on memory. The other studies in Netherlands, France, USA, or Europe not replicate these results. What happened in Latin America? In Latin America, Alicia Resende from Brazil showed the episodic memory improvement in illiterate adults attending the late life education, irrespective of low socioeconomic status. The FINGER's initiative in Finlandia was explored in Latin America, including 14 countries: Mexico, Dominican Republic, Puerto Rico, Colombia, Ecuador, Peru, Brazil, Bolivia, Chile, Argentina, and Uruguay. Screen participants 1,877 participants, and randomized 1,232 participants. The main aims from Latin FINGER include to investigate the feasibility of the FINGER multi-domain lifestyle intervention in Latin America context. The second objective is to investigate the efficacy of a systematic and multi-domain lifestyle intervention compared to a flexible lifestyle intervention on global cognition. Latin FINGER is the first non-pharmacological multi-center randomized clinical trial to prevent cognitive impairment in Latin America. A brief summary: the inclusion criteria is age 60 and 77 years old, they carry more than 6, or MSE, or CERAC learning task, or CERAC delay record score zeta, excluding dementia, major neurological or psychiatric disease, clinical condition, contraindicate intervention, basically physical activity. After screening, randomizations, and baseline evaluation include outcome primary, secondary, and exploratories, MRI, and APOE. The first studies on intervention include 2 years and classify 2 groups. A primary group of flexible lifestyle intervention that receive regular medical advice. Meanwhile, the systematic lifestyle group receive physical exercise, nutrition, cognition, and medical monitoring. The reevaluation is in the 12, 18, and 24 months. But the other funds is possible two researchers include two evaluations, 36 and 48 months, including MRI. So every participant will have a MRI in baseline, 24 months and 48 months. The primary outcomes include the Latam neuropsychological test battery. Secondary outcomes include processing speed, memory, executive functions, cognitive global and functional activities, and lifestyle questionnaire. And exploratory outcomes include cardiovascular risk, sleep habits, metabolic index, moods, quality of life, physical performance. And the intervention, the systematic growth is paulatine or month-to-month, starting with physical exercise, after nutrition, cognitive stimulation, and health coaching. It's important specify that intervention during 2 years and follow-up for 2 years additional include only evaluation with hoping that participants modify really lifestyles. This is the demographic and metabolic characteristics in Latin fingers baseline data include mean age 68 years, predominantly female, and the education is mean 12 years every country. However, Dominican Republic, Colombia, Ecuador, and Mexico include participants with low education, but there are scarce. The other characteristic of baseline data is the BMI plus 25 is 80%. What about intervention for dementia to communities for community health worker in Peru? This is an impact study. The impact study is a global health project funded by Britannic NIH, led by Jaime Miranda and Christopher Butler, with the objective to evaluate and help dementia screening tool and applied by community health workers. Community health workers are people in community with the proximity to every people in every block of home. This study is developed in Lima, capital of Peru, Tumbes, al north of Peru, Huancayo on the mountain, and Iquitos on the jungle. The first evaluation for community health worker include 32,000 participants. After participants will be classified in cognitive healthy, NCI, and dementia, including 3,600 participants. After the validation, include 600 participants and follow up at 6 and 12 months. The components on mHealth dementia screening tool in Peru include component 1, the consent informed, demographics, and ADI. The component 2 is the RUDAS. RUDAS is a brief cognitive screening validating to low education in different communities in Peru. Component 3 includes functionality to FEFE functionality activity questionnaires, and component 4 include comorbidities and others data. This is the step 1 to test and validation, the screening of health cognitive. According to different cutoff point of ADA, RUDAS, Pfeffer, the participants will be classified in cognitive healthy, mild cognitive impairment, and dementia, and include 3,600 participants with a study partner or caregivers. The observational studies is the next step, including evaluation of socio-demographic, CAT, labor and lifestyles, comorbidities, anthropometry, and fragility assessment. Step 3 include validation of breast cognitive screening applied by neuropsychologists from IPN, Instituto Peruano de Neurociencias, include gold standard, más digital cognitive assessment, clinical dementia rating, la neuropsychological evaluation based on Peruvian normative data of UDS, and TACA and TEL. In this subsample of a study, the collaborative dementia care via telephone on quality of life try include the ad patient and caregiver, 64 patients and 64 caregivers. After randomization, dementia expert providers, including healthcare workers, auxiliary of nurse, primitive healthcare facilities, and social workers, monitoring by community health workers, denominated in this study care team navigators. This the ad after recruitment and consent to realize outcome surveys in baseline 6 and 12 months. Basically include immediate techniques, medication, reconciliation and review, safety screen and recommendation, referral and caregiver education, caregiver well-being, behavior managing, and advising care planning. The proposal in the next steps is empowering the community health workers to alleviate the actual symptoms of dementia, but principal modify lifestyle of younger people is a study in process to obtain funds. What are the barriers to dementia risk reduction? Include external and internal barriers that influence opportunity for individuals to engage in dementia. The internal barriers include limiting resource, limiting modifiable dementia risk factors education, stigma, chronic illness or disability, lack of committing to behavioral change. Meanwhile, external barriers include inequality for LMC minorities, lack of social protection, reduced of some modifiable dementia risk factor, probably limitations to change the diet to Mediterranean diet is very expensive. Include root fruits, olive oils, and white fish. Very, very expensive, so difficult to adapt this possibility to modify lifestyles in these populations. Racial disparities and low socioeconomic status is other grand barrier in Latin America. In conclusion, a critical period for modifiable dementia risk factor intervention will be moved to young adults, ideal 18 and 38 years. The multi-domain intervention that subjects several risk factors and mechanisms simultaneously might be needed for an optimal preventive effect compared with single interventions. Latin Fringes is a first Latin American study, but not include participants with low education and low socioeconomic status in order to prioritize adherence. Include participants with socio-economic status from A or B, ideal to buy white fish, noodles, or olive oil that people with low socioeconomic status didn't buy. It's important to consider economical alternatives to nuts, olive oil, and white fish. Maybe in Mexico include the avocado or palta in Peru, other type of the oil in Ecuador and Colombia. Interventions in communities empowering community health workers is an alternative to provide brain health. Many thanks. I like to respond some questions or apply every topic. Thank you.
Gracias, doctor Custodio. Vamos a hacer una pregunta muy breve. Quisiera saber si el público tiene alguna pregunta para hacer. Bueno, dado que el público no tiene preguntas, yo tenía una pregunta para usted, doctor Custodio, y es, ¿qué particularidades ha identificado usted en la población peruana que podría funcionar como un estilo de vida que previene, digamos, como particularidad específica de la comunidad peruana?
Hey. Nuestros estudios en poblaciones nativas aymaras o quechuas del sur del Perú han demostrado la baja prevalencia de APOE4 en las comunidades aymaras. Tienen bajos niveles de presión arterial, diabetes y tienen un volumen cerebral, si bien es cierto, disminuido en tamaño, pero la capacidad de resiliencia permite que no tengan la probabilidad de tener deterioro cognitivo ni leve ni demencia. Nosotros atribuimos a que esto tiene que ver con la buena calidad de vida. No están en nuestras grandes ciudades bajo estrés, durmiendo muy poco, trasladándose de un sitio a otro largas horas para llegar a trabajar. Ellos solo tienen que despertarse cuando aparece el sol y dormir cuando se ve el sol, es decir, duermen mucho. Ellos caminan mucho para conseguir sus alimentos, no se someten a estrés, es decir, la calidad de vida de nuestra población aymara y quechua parecería ser un factor importante que cambia cuando ellos migran en situaciones pasadas del terrorismo en los años 80 y 90. Muchos aymaras y quechuas migraron hacia Lima, es decir, tenemos la misma carga genética de la gente que se quedó en los Andes y tenemos la misma carga genética en Lima y tienen otro tipo de manifestaciones clínicas neurológicas y más probabilidad de demencia, lo cual deja muy claro que el factor socioambiental es más importante que el factor genético. De tal manera que sí creemos que una modificación del estilo de vida basado en la mayor actividad física y la buena calidad de comida, sin incluir esta comida chatarra que nos gusta mucho en las grandes ciudades, son el principal problema.
Muchas gracias, doctor Custodio. De nuevo, invito al público a darle un aplauso al doctor Custodio y agradecerle su presencia el día de hoy.
Bien, muchas gracias.
For our final presentation of the day, we will watch a pre-recorded talk by Dr. Adolfo Garcia on language, AI, and global brain health. Dr. Adolfo Garcia is director of the Cognitive Neuroscience Center at the Universidad de San Andrés, Senior Atlantic Fellow at the Global Brain Health Institute, University of California in San Francisco, and Associate Researcher at the Universidad de Santiago de Chile. His work connects cognitive neuroscience, language, and brain health with a focus on multilingualism and cross-cultural research. He is the co-founder of Include, a global network that promotes cross-linguistic approaches to brain health, and the creator of Tell, a mobile application for speech assessment.
Thank you very much to the entire organizing team, and congratulations on this wonderful initiative. I will be talking about intersections between language, artificial intelligence, and global brain health. I will introduce the research landscape here, highlight some of the findings that we've produced in this space. Then I will briefly discuss our translational initiative rooted in the Tell app. And finally, I will address our efforts to tackle the diversity gap in the field and wrap up with some concluding words. When it comes to language and brain health, the first thing to understand is that we are living longer lives than ever before, but with this comes an unprecedented growth of neurodegenerative disorders, basically the number of persons with Alzheimer's and other forms of dementia and Parkinson's disease. Nowadays, they exceed 60 million people, and these figures are going to double in high-income countries and triple in low and middle-income countries by 2050. The methods that are used for standard diagnosis and phenotyping and monitoring of these disorders based on clinical interviews, paper and pencil tests, subjective reports from the patients and caregivers, in addition to neuroimaging measures, are limited in varied ways. They are expert dependent. They depend on high levels of expertise that are not equitable available across the globe. They are subject to human bias. Some of them are prohibitively costly, and neuroimaging, in addition, precludes participation of people who are claustrophobic or who have ferromagnetic implants, among other limitations. So all of this has spurred a quest for new markers. There's lots of excitement in the field of blood biomarkers and plasma biomarkers, but on the behavioral side of things, we are witnessing a surge of digital markers, and some people are trying to find signs of these disorders early, and even preclinical signs of these disorders in retinal scans, or using motion sensors, or even leveraging gamified versions of classical cognitive tests. And some people are trying to find clues into these disorders by analyzing aspects of naturalistic speech and language. And that's the subfield that we are operating on. We started working in this space over 10 years ago. And something that's interesting is that when one talks about neurodegenerative disorders of speech and language, all too often that intersection is narrowly conceived as referring to only primary progressive aphasia. And of course, in these syndromes, speech and language symptoms are the first and to be noted. They are the prime cause of disability and dysfunction in patients. But if one takes a broader view and dares to look at speech and language in disorders that are not primarily typified by speech and language disorders, one finds that these are no trivial domains. Basically, if you know which aspect of speech and language to focus on, you will find systematic deficits in up to 90% of persons with early stage Alzheimer's disease and Parkinson's disease, especially focusing on perhaps, for instance, lexicosemantic skills in AD, or motor speech measures in Parkinson's, and even in behavioral variant frontotemporal dementia, a syndrome that is sometimes juxtaposed to primary linguistic forms of FTD. Certain domains, such as lexicosemantic processing, can be impaired in 1 out of 2 patients. So there's really very, very good reason to analyze speech and language neurodegeneration with a transdiagnostic approach beyond the typical focus on PPA variants. And an interesting development here is that these digital technologies that I was alluding to, they are making their way into the clinical trial space even. Here we can see sort of the evolution of digital technologies in clinical trials in the US at least. And you can see that these have been a part of the game for the better part of 15 years now. But speech tracking, they are the newest kids on the block, and they're making an impactful contribution to the space. So when we started working on this, we had nothing. We didn't have a single line of code. Everything was to be built. And we made some breakthroughs in this framework that I want to introduce. The first thing that we did was we were able to standardize and validate a collection of speech elicitation tasks, including spontaneous, semi-spontaneous, and non-spontaneous speech elicitation tasks, which vary strategically in their motoric, cognitive, and even affective demands. From each of those recordings that we obtain, we first have the audio signal, the recording proper, from which we can analyze many, many aspects that have to do with speech rhythm, with pitch changes, with voice quality, with intelligibility, with articulation, and several other things. But also we open a second realm of analysis. We transcribe those audio files, and now we have text. And from those texts, we can use natural language processing algorithms to analyze aspects, hopefully markers, that pertain to the lexical or the semantic or the morphological or the syntactic or the topological properties of the pieces of discourse that are produced. And all of these features we can feed into machine learning pipelines to perform a series of analyses that will allow us to detect how robustly or not these features can identify whether a person has a certain disorder or doesn't, whether a person has a certain phenotype of a disorder, or how, using machine learning regressions, for instance, whether those features are predictive of the severity of their core symptoms, or even of underlying patterns of neuroanatomical or neurofunctional abnormalities. So with this approach, I'm going to show you a sampler of some of the things that we've done. This is a paper that we did in non-fluent variant PPA. This is a form of aphasia that's typified by early alterations in motor speech capacities, sometimes accompanied by agrammatism. But here we had participants engage in a very simple task, which consisted in reading a text out loud. That's all it took. And we focused on speech timing metrics. So basically what we are measuring is the number of pauses and the duration of those pauses as they read, the number of phonated segments and the duration of those phonated segments, and even more interestingly, perhaps, some calculations therefrom, including in particular articulation rate, which is basically a measure of how much speech signal there is in a span of 1 minute upon subtracting silence segments. So with this metric, we found that people with non-fluent variant PPA, as we predicted, they have a significantly lower articulation rate than do healthy controls. But also, this measure allows us to find significant differences relative to another PPA variant, here the semantic variant of the disorder. Those deficits in the non-fluent variant PPA cases, they correlated significantly with the volume of motor, premotor, and supplementary motor cortices, as well as the volume of the inferior frontal gyrus, all in the left hemisphere. And with machine learning analysis, we found that this metric allowed us to classify persons with non-fluent variant PPA from healthy controls with an area under the curve of 0.95. Now, the most interesting part of this study is that non-fluent variant PPA can actually follow from different underlying pathologies. But the way to ascertain the underlying pathology in these disorders is basically to wait until the patients pass away, so that then you can perform analysis of pathological analysis post-mortem on brain slices. And the patients in this study had unfortunately passed away, but they had very generously donated their brains. So what we did now was we went back to the recordings, and it just so happens that non-fluent variant PPA can be underpinned by two different pathologies, maybe more than that, but the most two most typical ones are corticobasal degeneration and progressive supranuclear palsy. And the interesting thing is that even though the syndrome, the behavioral manifestations may be similar, the course, the trajectory of the disorder is going to be quite different depending on that underlying pathology. So what we did was we went back to the recordings which had been obtained nearly 10 to 15 years before the patients passed away, and we tried to use our automated speech metrics to see whether we could identify the underlying pathology, and we were able, to our gracious surprise, to do so with an area under the curve of 0.82, which suggests that there is information in a very brief 1-minute ecologically valid task that can predict pathological patterns that nowadays can only be ascertained after death. We've also worked with Alzheimer's disease, and we came up with a new approach to analyze data from verbal fluency tasks. These are tasks in which participants are asked to produce as many words as they can in the span of a minute, following either phonological or semantic cues. In 99% of the studies, what's done is that clinicians count the number of valid responses, so how many words were produced that are task compliant, basically. But this, while this is very sensitive to Alzheimer's, it's very unspecific, because this approach will also yield deficits in people with other forms of dementia, and actually with many other neurodegenerative, neuropsychiatric, and even neurodevelopmental disorders. And also, this leaves you blind as to which areas of semantic memory are most affected. So we came up with a different approach in which we take all the words that participants produce. We don't really care whether the words that they produce are task compliant or not, and we analyze them in terms of objective properties. We extract information, features that have to do with how frequent the word is used in daily language usage, their length, their number of phonological neighbors, the familiarity, how conceptually precise or granular they are, and so on. And all of this information we feed into machine learning pipelines similar to the one that I showed before. And we were able to show that by combining these features, we can identify whether a person has Alzheimer's dementia or not, with an area under the curve of 0.89. And when we replicate the same approach on people with behavioral variant frontotemporal dementia, which is another form of dementia that is sometimes confounded with or difficult to disentangle from Alzheimer's, we find results that are slightly above, which is good, because this means that this approach may have information that may contribute to differentiations between syndromes. Some of these features that we analyze, they correlate significantly with executive dysfunctions exclusively in Alzheimer's, not in BVFDD. And they also map onto syndrome-specific patterns of brain atrophy and functional hypoconnectivity along canonical brain networks, again, exclusively for Alzheimer's dementia. We then replicated this approach, very similar to what I just showed you, adding a few features in people with mild cognitive impairment, which is a form of cognitive decline that does not compromise autonomy, but that can be clinically measurable, and it increases the chances of developing dementia substantially. Basically, we did the same thing using the word properties that I mentioned just now, and the speech timing features that I mentioned in the non-fluent variant PPA study. And I would like to draw your attention to panels C and D here. In panel C, what you see is that when we combine the word property and the speech timing metrics, we can classify between people with MCI and healthy controls with an area under the curve of 0.80. And the interesting thing is that when we use the very same machine learning pipeline, but now using as input features standard cognitive tasks that are used to diagnose these disorders, including measures of episodic memory, attention, cognitive flexibility, and even the count of valid responses in verbal fluency tasks, we reach an area under the curve of 0.72, suggesting that the information that we can glean from this automated speech and language analysis can actually contribute and hopefully improve the identification of people with this particular disorder. And some of these features, they also map on brain volume metrics in the population. We've also worked with Parkinson's disease, something that we've reported quite often using more standard paper and pencil and computerized tasks, is that people with Parkinson's disease have distinct difficulties comprehending, associating, and producing action words. So even in single word, single sentence, or text level studies, we showed that this particular semantic field is distinctly challenging for people with Parkinson's disease. But until this paper that we put out in 2023, no one had automated a measure of this particular domain. That's what we did. We asked participants and healthy patients and healthy controls to first read an action-laden story and then retell it in their own words, and also to read a non-action-laden story as a control condition and to retell it in their own words. And basically, we then identified all the action verbs and the non-action verbs in those original stories. We framed them as semantic seeds, if you will, calculating vectors and embeddings from them. And then we estimated the distance between the words produced by the participants in the retellings and those semantic seeds. And what we found was that the distance relative to the action concept seeds were highly discriminative if you focus on panels B and C here of people with both Parkinson's disease patients without mild cognitive impairment, but also Parkinson's disease patients with mild cognitive impairment, suggesting that this particular metric may be sensitive to Parkinson's irrespective of the cognitive phenotype that the patient may present with. We have additional evidence on more general aspects of semantics in Parkinson's disease, but in the interest of time, I'm going to go very quickly over this. We found that people with Parkinson's disease, when they have to describe concepts, they favor more concrete, more imageable, and less semantically variable features in their descriptions, and that some of these features map onto functional connectivity disruptions in PD. Now, we are also interested in making sure that this basic research that we produce in the lab is not confined to the lab. So we created a company and an app, both of which are called TEL. And this is a startup that we built to try to take all this information, all these features, all these findings into actual clinical settings for direct use by clinicians and for direct report to patients and their families. So we've secured investments from two life science funds. We have created this growing ecosystem with clients and incubators and accelerators and partners from different fields across the globe. We have different data acquisition modalities, and we have different combinations of tasks and features that are informative of domains that are relevant to these disorders, including assessments of motor speech, semantic memory, episodic memory, and emotional processing. And also, we are interested in making sure that the information that we glean with this technology can be easily interpretable by clinicians and patients, so that we are working hard on validating visualizations that can be readily apprehensible by these stakeholders. But also, we are interested in tackling a challenge that has to do with diversity. In the world, there are around 8 billion people, perhaps over 100 billion people if we believe a recent paper that was published in a very prominent journal just 2 weeks ago. And if you take a look at all those people and you put together all of those who speak English to any level of proficiency, native speakers or non-native speakers, they would represent 17% of the world population. The problem is that research on speech and language markers of neurodegeneration is tremendously biased, because 75% of the studies out there are based on English-speaking cohorts. So a language that's spoken by only 70% of the world represents over 75% of the evidence out there. This is inequitable. We don't know much about the other over 6,500 languages in the world. And we are working deliberately on tackling this challenge by making sure that TEL is used in different hospital clinics and research institutions in different continents with different socio-cultural and ethno-racial profiles. We are also working in the Red Lab Consortium with this beautiful project that we are running with Drs. Ibanez and Gorno Tepini, leveraging TEL to find the most robust automated speech and language markers of dementia in Chile, Argentina, Peru, Colombia, Mexico, and the US. And we have also this transcontinental project that we are running with dear colleagues, Drs. Borgesani and Soto Aniari, spanning cohorts from Switzerland and Peru who are bilingual. And also we created the Include Network, which is the largest network devoted to cross-linguistic research on brain health. We are now 150 members strong, with members from 90 countries and over, 90 centers and over 25 countries. And we are working on pre-existing data from nearly 5,000 patients in different projects aimed to understand which of these speech and language markers may be generalizable across typologically different languages and which ones may be more language idiosyncratic. And also we started this dissemination effort, a webinar series hosted by the Alzheimer's Association, focused on language diversity in brain health. The next rendition will be on April 22nd, based on focusing on the adaptation of cognitive tests across languages and cultures. So please join us if you find this topic to be of your interest. So to wrap up, this is a very exciting field. It's thriving, but it also has core challenges. We need more research on larger samples. We need longitudinal studies. There are very few of these. We need more linguistic and sociocultural diversity. And we also need to work concertedly on efforts to harmonize the tasks and the approaches that we are using for data collection and analysis. So while we seek the final word on neurodegeneration, let's work on the words we have now. So thanks on behalf of the wonderful team that we have at the Cognitive Neuroscience Center at Universidad de San Andrés and our network of collaborators nationally in Argentina, regionally in Latin America, and globally across the world. Thanks for your attention, and I wish you a fantastic rest of the conference.
Thank you so much, Dr. Garcia. I have two questions of this topic. In first place, Dr. Garcia, we know speech varies widely across culture and languages. How can real-time speech analysis like speech tracking help us understand these differences?
Yes, it's one of the main challenges. So the important thing, one of the most important things is to always have data from healthy controls from each of the dialects or regions or countries that you're working from, because what really matters is not so much the speech measures themselves, but rather the degree of deviance from the output of patients relative to the output of socio-demographically matched controls. So by relying perhaps on weighted scores or z-scores that are normalized relative to benchmark data from the same population or region, that's what really matters. And this is an important point because the types of words, the grammatical constructions, the prosody that typifies not only different languages, but even different variants of the same language. If you think about the wide spectrum of Spanish variants across Latin America, right? I mean, if you go from Argentina to Chile to Colombia to Venezuela to Peru to Mexico, it's very, very different. So we really need to work hard at making sure that as we are building our patient cohorts from which we extract these data, so do we build reliable and comparable healthy control cohorts so that we can start working on is the differences between both rather than just the face value of the measures that we derive directly from the patients.
Okay, thank you so much. And the second one is speech tracking has transformed how we study the brain and the language. In your view, what's the biggest technical challenge for real-time speech tracking in clinical or research today?
So yeah, we do this in real time, but we do this in a controlled fashion. So what we've learned is that it's not just measuring speech as it comes. So if you were able to go over the ethical hurdles of recording someone, 24/7 with, I don't know, Alexa, or by, monitoring their phone or something, that might be great, but it may not be the best thing, because what really matters is that you expose participants to tasks that aim at forms of discourse production that are eloquent, that are relevant. So the tasks that we use, not all tasks are good for all disorders. So for instance, a paragraph reading task is very, very, very useful to capture measures of hypokinetic dysarthria, for instance, or apraxia of speech. And that way, they are very relevant to working with people who have motor speech disorders, non-fluent variant PPA, Parkinson's, Huntington's, spinocerebellar ataxia. But if you want something that's a better proxy of semantic or episodic memory, then you would probably need a different type of task, right? So we do real-time measures, but in a very, very strategic way, I would say. The main challenges that they're manifold. How to deal with the heterogeneity of the audio signal across different acquisition devices. So some of the protocols that we run, we have the fortune to be able to standardize and harmonize everything and make sure that all the sites are using the same microphones, the same tablets. We are trying to keep mic to mouth distance constant and the background noise levels also controlled. But sometimes in more naturalistic settings, this is not possible. And accounting for that variability, we have ways of doing it, but it always comes at a cost of signal quality and signal quantity, even. Another thing has to do with the standardization of tasks, and actually perhaps even moving beyond the tasks that for historical reasons have become sort of the go-to stimuli. Everybody keeps relying on the cookie theft picture just because it's the recordings are out there, but it does really, it may be one of the very worst stimuli out there. It's not culturally relevant for many populations. It's dated, it's black and white, and it probably does not have the richness that more dynamic stimuli such as videos can provide. And then another challenge is, I will mention one more. I worry that there's one particular data set that's being made publicly available, the address, and then another version of it, the address of data set, both based on the PIT corpus, and it's publicly available. And you see papers and papers and papers and special issues based exclusively or primarily on this data set. And I worry about that. On the one hand, because the diagnosis of AD in that data set, they predate the 1984 McCann criteria, and they're actually just based on MMSE cutoffs. The quality of the audios is dreadful, I have to say, and everybody keeps drawing patterns from that. And I fear that we will be sort of overfitting our conclusions about the field to a single data set. And that's never a good thing. So increasing the diversity of data sets and making sure that that data sets that are good quality and informative and diverse are made publicly available is another of the, I don't know if technical, but certainly methodological challenges that we still need to meet.
Okay, thank you so much, Dr. Garcia. It was a pleasure having you at this event and learning about your work. We look forward to continue to collaborate with you and your team in the future. So with this, we can finish.
Thank you very much. Again, wonderful event. Thrilled to have been a part of it and looking forward to more interactions with you and the entire team. Thank you.
For sure. So.
Thank you at the distance at Dr. Garcia, and thank you to Carolina for the recording, and thank you all for and the remaining ones for your participation at day one of the Colombian Brain Health Congress. Tomorrow we'll have the beginning of our session at 8 a.m. in the Jaime Hoyos Auditorium. It's the auditorium that you go next to this road and just keep going until the university finishes. And that's the last building that's in the basement, the auditorium that we have tomorrow. And the later, the Wednesday. So we'll hope to see you there. We'll leave you with a short comments and observations survey for the ones of you that wants to leave comments about the Congress, about the panelists. We encourage you to reach out to them for questions, for projects, and thank you very much for attending today.
Have a pleasant evening, and we look forward to seeing you tomorrow. Thank you.
See you tomorrow. Tenemos el auditorio hasta las 5:40, así que creo que está lloviendo, así que no hay problema.